To continue talking about our urinary system, we'll go ahead and talk about the nephrons. So as we said, the nephrons are made out of the renal corpuscle and the renal tubules. We're going to go ahead and zoom in on the renal corpuscle to see what it's made out of. The renal corpuscle of the nephron is made up of two parts. It's made up of a hollow cavity known as the glomerular capsule, or you can call it the Bowman capsule. And inside of that capsule, inside of that hollow capsule, there is a bunch of tangled up capillaries. We refer to those capillaries as the glomerulus. Now, the capsule, the glomerular capsule or the Bowman's capsule, it's made up of two layers. The superficial layer is known as the parietal layer, which is made up of a simple squamous epithelial tissue. And then the visceral layer is actually internal right here. That yellowish layer that you are seeing is the visceral layer of the Bowman's capsule. That visceral layer, it's made up of some modified branching epithelial cells known as podocytes. So those, again, that yellowish layer that you are seeing, those are the podocytes. And podo meaning feet and cyte meaning cells. So those podocytes, the reason why they call them feet cells, because they have extensions, as you can see right here, those extensions. And those podocytes, they go ahead and they cover up the glomerulus. They cover up those capillaries. Again, the capillaries are the glomerulus. So what's the importance of having the visceral layer, those podocytes right on top of the glomerulus? Since the glomerulus are tangled up capillaries, we know that capillaries are porous. And so those glomerulus capillaries, they have a bunch of pores. And so when the blood enters into the glomerulus to get filtered, as we said, the first step to make urine is filtration. So as the blood is entering into the glomerulus via the afferent arteriole, the blood is going to get filtered. And so therefore, since the glomerulus is pretty thin and porous, small solutes are going to be able to pass through. The podocytes put restriction to prevent large things such as your cells and your large proteins from passing through. So that's the importance of having those podocytes. It puts a restrictive layer over the glomerulus. Now, if your cells and large proteins are not able to pass through the glomerulus, where are they going to go? Now, they are basically going to exit back out via this arteriole right here, known as the efferent arteriole. So, one more time, the blood is going to enter into your afferent arteriole. It's going to enter into the afferent arteriole, and it's going to, the blood is going to pool right here in the glomerulus, which is basically tangled up capillaries. Those capillaries are thin and porous, therefore they're going to allow small solutes to be able to pass through. Things like water, glucose, small ions like sodium, potassium, etc. Even waste is going to be able to pass through the glomerulus, entering into the hollow cavity, and from there, those solutes are going to enter into the proximal convoluted tubule. Proximal meaning close by, so it's close to the glomerulus. Convoluted meaning twisted. So the solutes that are able to pass through the glomerulus will enter into the proximal convoluted tubule. Anything in your blood that was not able to pass through the glomerulus, because they are pretty huge compared to the water and compared to the glucose, etc., such as your cells and your large proteins, will exit back out via the efferent arteriole. From there, it's going to get drained into your small veins known as your venules, and then from there it's going to actually exit your kidney completely via the renal vein. So if we go back to this image right here, if you remember, we are right here, okay? So we are up here in the renal cortex and medulla region. The blood is going to enter into the glomerular capsule via the afferent arteriole. This capsule contains the glomerulus, which is basically a bunch of capillaries. You can think of the glomerulus as a coffee filter. When you make coffee and you put that filter paper, not everything is going to be able to pass through that filter paper, right? Small things like the water and the small, like, coffee flavor is going to be able to pass through the filter, while the large coffee beans are not going to be able to pass through. So same thing with your blood. The blood contains both small solutes and large cells and large proteins. So as the blood enters into the glomerulus, the small solutes and the small ions and the fluids are going to be able to pass through. The large cells and the large proteins are actually just going to exit back out because they cannot pass through that filter, through the glomerulus. So they're going to exit back out via the efferent arteriole. So the nephron is made up of that renal corpuscle, which, as we said, it's made up of the capsule and the glomerulus. The nephron is also made up of a bunch of tubes. That very first tube right here connected to the renal corpuscle is known as the proximal convoluted tubule. And again, you can see how twisted it is, therefore it's known as the convoluted and proximal because it's close to the glomerular capsule. So whatever is going to pass through the glomerulus, we're not going to call it urine. We are going to call it the filtrate. So the filtrate is going to leave the glomerulus and leave the capsule and enter into the proximal convoluted tubule. The proximal convoluted tubule is made up of a simple epithelial tissue. The cells are cuboidal, so it's a simple cuboidal epithelial tissue. And on the apical surface of those cuboidal cells, there are a bunch of microvilli. The microvilli are going to increase the surface area and increase the absorption. So that filtrate that's going to enter into the proximal convoluted tubule contains a lot of nutritious things like glucose, and it contains a whole bunch of ions that are important to your body to retain. It contains lots of nutritious elements, and so you do not want to lose all of that filtrate as urine. So the second step of urine formation, which is reabsorption, is going to take place within your proximal convoluted tubule. Almost 65% of the filtrate is actually going to be reabsorbed out of your proximal convoluted tubule. And why is a huge amount of that filtrate is going to be absorbed there within your proximal convoluted tubule? Because there's a bunch of microvilli on the apical surface of your proximal convoluted tubule. Now, after the filtrate passes through the proximal convoluted tubule, and we're going to reabsorb almost 65% back, the filtrate, whatever is still going to be left in your proximal convoluted tubule, that filtrate is going to make its way into the nephron loop. So that loop right here the filtrate is going to make its way into that loop where more reabsorption is going to take place. So if there is still some water or some pretty important ions or glucose, they are still going to be able to be reabsorbed out of your nephron loop. Then whatever filtrate is going to be found within your nephron loop, that filtrate is going to make its way into the distal convoluted tubule, where more reabsorption can take place. But the distal convoluted tubule, again made out of epithelial tissue, simple epithelial tissue, the cells within your distal convoluted tubule don't have as many microvilli as what you had on the surface of your proximal convoluted tubule cells. And so therefore, the amount of reabsorption that's going to take place within your distal convoluted tubule is not going to be as high as the reabsorption that's going to be able to take place within your proximal convoluted tubule. Now, when we say reabsorption, that means the water and the ions, etc., are going to be exported out of the tubules, and they're going to enter into a bunch of capillaries that are going to be right on top of your tubules, which we will talk about their names. So you reabsorb those ions and the water, etc., out of those tubules, and they're going to enter into a bunch of capillaries, and from there the capillaries will drain all of those important ions back into your bloodstream. Now, whatever filtrate is going to stay within your distal convoluted tubule will go ahead and it will get drained into your collecting duct, where more reabsorption can also take place within your collecting duct. So within your collecting duct, still ions and water, etc., can go ahead and get reabsorbed out of the collecting duct back into your bloodstream. Therefore, we keep referring to it as the filtrate and not urine, because as you can see, we reabsorb almost total. We are going to reabsorb a total of 99% of our filtrates, where we take them out of the tubules and send them back into our bloodstream. So the first step of urine formation is filtration, which will only happen within the glomerulus. The second step is reabsorption, where you are reabsorbing almost 99% of your filtrate and you are returning it back into your bloodstream. And reabsorption can happen within the proximal convoluted tubule, the nephron loop, the distal convoluted tubule, and the collecting duct. Now, the third step of urine formation is secretion. Now, as we are reabsorbing things out of our filtrate, waste could actually be reabsorbed back. And so therefore, secretion is where you're going to selectively take the waste or certain ions out of the bloodstream back into the tubules. Secretion can happen anywhere within your tubules, including the collecting duct. By the way, that collecting duct right here, notice how it has so many branches. That's because many nephrons can drain the filtrate into one collecting duct. And again, all of this is up here. Now, there are two types of nephrons we have within our kidneys, cortical nephrons and juxtamedullary nephron. Cortical nephron, the reason why it's called cortical, because it's closer to the renal cortex. So if this is the renal cortex and this is the renal medulla, The cortical nephrons are closer to the cortex region. The juxtamedullary is closer to the medulla region. Juxta actually means close to, so they're close more to the medulla region. So this image shows you as the blood is entering into the glomerulus via the afferent arteriole. The glomerulus is going to go ahead and start filtering the blood. Small things that are able to pass through will enter into the tubules. Large things that are not going to be able to pass through the glomerulus will exit back out via the efferent arteriole. From there, whatever is going to exit back out via the efferent arteriole, it's going to go ahead and it's going to get drained into those purple tubings, which are actually a bunch of capillaries known as the peritubular capillaries. Peri meaning around. So it's going to get drained into those peritubular capillaries, and from there it's going to get drained into your small veins, which are up here, and eventually those veins are going to keep merging until it's going to exit your kidneys via the renal vein and back into your inferior vena cava. Now, we said that within your proximal convoluted tubule and your distal convoluted tubule, reabsorption will happen, where ions and glucose and water will exit the tubules. Now, where are they going to go? Those things are going to exit out of your tubules and enter into the peritubular capillaries, and from there, the ions and the water are going to also one more time get drained into the veins and eventually exit your kidney via the renal vein. Now, the nephron loop, we said that reabsorption can also happen within your nephron loop, where the ions and the important things like water and glucose will be reabsorbed out of your nephron loop, and it will enter into a bunch of capillaries surrounding your nephron loop within your juxtamedullary nephron, and those capillaries are known as the vasa recta. And again, the vasa recta is going to take all of that important things that you've just reabsorbed, and it's going to drain it into your venules. Venule just means small veins, by the way, and then it's going to exit your kidney via the renal vein. Any secretion that will take place is basically anything that was reabsorbed back and now it's within the peritubular or the vasa recta. It can be selectively and actively transported out of those capillaries back into the tubules. Whatever is going to be found within your collecting duct and no more reabsorption takes place will eventually become urine. So this image right here just kind of shows that as the blood enters into your renal artery and the renal artery starts to branch out, by the time we get up here to the renal cortex, the blood enters into those very small arteries that are going to be located up here, known as the afferent arteriole. That's where the blood is going to enter into the capillaries known as the glomerulus, and this is where filtration will take place. Whatever is not able to pass through the glomerulus, remember, kind of like our coffee filter, whatever is not going to be able to pass through the glomerulus, it's going to exit out via the efferent arteriole. From there, it's going to drain into the capillaries, whether the peritubulars or the vasa recta, and it will make its way out of the kidney via eventually the renal vein, and it's going to get drained back into the inferior vena cava. whatever is able to pass through the glomerulus will take a different route. It's not going to exit via the efferent. It's going to enter into the proximal convoluted tubule, and then the nephron loop, the distal, the collecting duct, and eventually down the ureter. Whatever is going to exit the ureter, we are going to call it the urine. So the urine is going to eventually exit the ureter. From there, it's going to go into the urinary bladder and out of your body via the urethra. So our kidneys process 180 liters of fluid every day, but because we reabsorb almost all of that filtrate back, we are only going to release 1.5 liters of urine. So first we filter our blood, then we reabsorb almost all of the filtrate back, and then if there are still some waste within your peritubular capillaries or your vasorecta, they will be pushed out of those capillaries into the tubules to be secreted as urine. Filtration only happens within the glomerulus, as I said. Things like cells and proteins will not be able to pass through, and so they will exit right away via the efferent arteriole back into your bloodstream. As I said, we reabsorb almost 99% back of our filtrate, and then we can selectively move substances out of our blood, so out of our peritubular capillary or the vasorecta back into the tubule. Whatever is going to be found within your collecting duct and no more reabsorption is taking place is going to start leaving your collecting duct, and it's going to eventually drain into the ureter and from there into the bladder and out of your body. So this just summarizes one more time the 3 steps. So the blood enters into the afferent arteriole. It's going to pass into the capillaries, right, which are your glomerular capillaries. Small things such as water, ions, drugs are going to be able to pass through the glomerulus, enter into the tubules. Almost 99% of that filtrate that will pass into the tubules will actually be reabsorbed back into your other capillaries, the peritubular and the vasorecta, and from there it's going to go into your bloodstream. Secretion is where you selectively take things out of the peritubular or the vasorecta and push them back into the tubules. And eventually, whatever is going to be pushed back into the tubules and it's not going to be reabsorbed one more time will become urine. Anything that's large and it's not able to pass down the glomerulus in the first place will just exit back out via the efferent arteriole. From there, it's going to drain into the peritubular or the vasorecta and exit into your veins and eventually completely exit the kidney via the renal vein and will be drained back into your bloodstream.